Hi everyone, I'm Jiajing and I'd like to extend a very warm welcome to SynBio's very first webinar of the new academic year. Thank you very much for joining us and I hope you're very excited to hear today's talk from Professor Leonardo Morsut. If this is your first time joining us, SynBio.Oxford is a student-run society that aims to provide an educational and outreach platform for synthetic biology. We do so by bringing you workshops and talks from both academia and industry so you can learn more from their knowledge and experience. And this is all possible thanks to our sponsor, Integrated DNA Technologies, or IDT. So today's webinar is titled Learning to Program Tissue Development with Artificial Genetic Circuits. And we're very honored to have Professor Leonardo Morsut with us today. Professor Leonardo is an assistant professor of stem cell biology and regenerative medicine and biomedical engineering at the University of Southern California. Having been born and raised in Italy, he began his career in the US in 2012 as a postdoctoral scholar at the University of California, San Francisco. There he developed a new class of synthetic receptors called synthetic notch receptors that have since been proven to have far-reaching implications for cell therapies for cancer, autoimmunity, and regenerative medicine. In 2017, he started his own lab in USC, focusing on using synthetic biology approaches in multicellular mammalian systems. The goal of his lab is to find ways to build smart designer tissues with user-defined structure and function. Now, before we begin the talk, I'd like to mention that today's webinar will be recorded, as all our webinars will be published on our YouTube page. Please also note that we will have a Q&A session after the talk, so if you have any questions during the talk, feel free to type it into the chat box and we'll address them in the Q&A session. Now that's all from me. Over to you, Professor.
Amazing. I'm excited to be here. Talking to a crowd of people that are all excited about synthetic biology. It's particularly exciting for me. And we all like to think about this idea that we can engineer and control cells, control biology. It's a great starting point because I think it's going to be really impactful in the future of humankind, if you want, and society and technology. I want to start by focusing. I know that we are all excited about synthetic biology. I want to focus just a quick at the beginning, plant a seed of the side the side of the coin is more about biology. And I just want to focus our attention to the fact that at least one way to think about it is that synthetic biology is cool, is exciting because biology itself is pretty exciting. It has had millions of years to develop and to evolve machines and to evolve genetic circuits, and what it can do is pretty amazing. And the reason why we are excited about controlling it is because biology itself is a very, very powerful, if you want, is a very powerful technology for building machines, if you want. And so you see here, you have animals, plants, humans are all, if you want to take a look at that, like, from that perspective, they're all machines. They perform very, very complicated tasks. And so my biggest fascination in biology is how are these machines built? So let's say you take a seed of an orange tree and you plant it, and in a process of development you construct a machine is constructed, which we call a tree, an orange tree. And the orange tree is a machine that if you give sort of water and a little bit of nutrients from the soil and a little bit of sun, this machine is able to build oranges with these very, very simple starting materials. And so that's the orange tree. But you can think of a brain, you can think of your favorite piece of biology. And when you look at it, the question then for me becomes, how does nature build these complicated machines? And the answer is in this next slide. This is a time-lapse movie of embryonic development. starting from a single cell and going all the way to an organism. In this is one of my favorite videos. You see here is the embryonic development of a zebrafish. This is a small fish with stripes that we call zebrafish. And I'm going to show it again as I talk a little bit more about this. This is as a building technology, development takes a single cell into a fully functioning organism. And you see, in plants you start from a seed. In animals you start from a fertilized oocyte. It's this process of building with cells. It takes this form of starting from a simple material, and it brings about a trajectory, a developmental trajectory, and it builds during which it builds the machine, the final machine, the adult machine that then performs all these interesting functions that we like to see. And so, for me, in my lab, in my career so far, I've been driven by this aspiration of learning what are the design logic of this technology that is embryonic development, that is a genetically driven technology that is able to control the process of development from a single cell to a fully functioning machine. And so, if we can schematize my dream sort of goal that I'm trying to achieve with my research, would be being able to take a cell, introduce artificial genetic networks, artificial genetic circuits made of cell communication, made of promoters, made of receptors, and those circuits would drive the patterning, the morphogenesis, the cell division, and eventually the differentiation and the functional formation of a structure that's new, that's novel, that wasn't there before, that the cells wouldn't do by themselves, but they would do because we have introduced these artificial genetic circuits. And so this is sort of the vision and the sort of the moonshot that we have in mind, and that guides a little bit of everything that we do in the lab. I put there a few of the reviews or articles that we've written. It's more like conceptual on the latest advancement of this field that some people call synthetic development or synthetic morphogenesis or tissue development engineering. We are not the only ones, and just want to say that. And this is sort of a new approach towards using the synthetic biology principles for developmental systems and morphogenetic systems. So we are not there yet. We haven't been able to build sorry, I've been signed out from Zoom. Do you still hear me?
Yeah, we can still hear you and see your screen, but it's not the presentation at the moment.
Right. Let me see if I can fix it quickly. Is this better?
Yeah, okay. The presentation's back.
All right, thanks. So we were saying that we're not there yet. We haven't built a fully functioning worm or a fully functioning organism driven by genetic circuits. But we started building a few of the pieces. And so what I'm going to show today are some of the pieces that we have. And I'm hoping I hope to get you thinking about what could be things that you are either interested in getting from the types of tools that we develop that you can use in your research or for your deepening your knowledge of this field, or maybe you get excited and you want to participate in this effort and you want to see if you want to join this lab or other labs that are pursuing these types of efforts. And so today I'm going to show you how we've used the synthetic signaling of the synthetic notch kind that was mentioned earlier by Gia to drive some form of symmetry breaking and patterning in spheroids. How then a second part in which we're going to show you how we have a mechanochemical coupling in a specific type of patterning circuits, and then finally how we can pattern cell differentiation, that we've been able to geometrically control cell differentiation thanks to signaling from the ECM to the cell as opposed to cell signaling. And I'd like to start to finish this intro by showing the lab slide. This is a rather old lab picture, but we are established in Southern California, in Los Angeles, and we are in the Department of Stem Cell and Regenerative Medicine, and we try to do this, divide our efforts in these three sort of more specific subfields. We build these toy models of synthetic development. We collaborate with people in the department that are doing more organoids type of work. Organoids are organ-like structures that are built from stem cells, with originally with no synthetic biology at all, just with growth factor in the media. But we are introducing, we think that synthetic biology controls can help their reproducibility and the control of the organoids development. So we are collaborating with a few people in the department to do that end. And we are also keeping the sort of translational ideas and projects and potential of these technologies alive with project where we engineer cells with the idea of using them as therapies for degenerative disease. Although today is going to be mainly about toy models. Can you confirm that you see the slides and see the movie moving? Yeah, okay. Okay, so, and yeah, as Gia said, feel free to ask questions, put them in the chat if something is, and yeah, if something is not clear, you can even interrupt me, as these are going to be three different sort of stories. I'm gonna move fast over these as this was a longer presentation. Yeah, I wanted to give a sense of the synthetic pathway that was mentioned as just a little bit of explanation of what it is. So the synthetic pathway that we developed is inspired by an endogenous pathway that cells use during development. So cells, to form the patterns that they form during development, they extensively communicate with one another. And one way that they do so is by some cells expressing a signal and other cells expressing receptors that read the signal and change their behavior accordingly. And one of such cell-cell communication pathways is called Notch, and the ligand is called delta that you see here. On this, the delta is expressed on the membrane of the sender cells, and the receiver cells produce the Notch receptor. And when the two bind, there's a cleavage event that happens in the transmembrane domain, which liberates the intracellular domain of the receptor, which is a transcription factor that goes in the nucleus and activate for Notch target genes. And this is the naturally evolved Notch signaling, naturally evolved ways that nature came up with this idea of making cells communicate with each other in these ways as a contact dependent. The two cells communicate only if the two cells are in contact. So we were very much inspired by this way of communicating that the cells have. And so we made it into a synthetic version by pretty much replacing the extracellular domain with an antibody-based recognition domain. They now can recognize your favorite target gene. It's an antibody based, basically, and so there can be an antibody against GFP, can be an antibody against something that the cells don't ever encounter, like GFP, mCherry, other synthetic ligands. It can be an antibody that recognizes a normal antigen, a normal membrane protein like CD19, or other examples of tumor antigens or disease-specific antigens. And it doesn't bind to delta anymore. It retains the capacity of when binding the ligand to induce a cleavage of the transmembrane domain. And so this is the sort of the part that we have maintained from the endogenous notch receptor. And then we put a artificial transcription factor as an orthogonal transcription factor, different kinds. It can be GAL4, can be TTA, can be zinc finger based. And other people have started to put other kinds of artificial transcription factors. They can then, when the receptor is engaged, this can be again liberated from the membrane, you go in the nucleus and activate for whatever target gene you have put in. As this is receiver cell is engineered with two parts, the receptor and the target genes. You augment these cells with these two components, and when they are in, the synthetic receptor can activate whatever target gene you have put downstream. And so with this, you can start to encode cell-cell communications, pathways that are user-defined, and that the cells don't, in a way that's orthogonal from what the cells are already doing. So you can interface with cell behaviors, as you'll see. And so so the majority of what I'm going to show is going to be using SynNotch receptors, different kinds. I do want to say that SynNotch is not the only synthetic receptor that is used in mammalian cells. Actually, it's just one of many. We're coming out with this review where with the Leonard lab, where we are talking about the evolution of synthetic receptors, and we have this nice figure that highly put together. Where if you look at SynNotch, it's just this little guy on the left here. So there are many other synthetic receptors. They've been mainly developed with the idea of using for cell therapies, for controlling cell behavior in cell therapeutic approaches. So no other synthetic receptor has been used for synthetic development yet. But the idea is that in the next years, there's going to be a lot of possibilities for using also other receptors that are not SynNotch. As during development, cells communicate not only with notch, they communicate also with other kinds of communication pathways, or some are more long range, and some are more hormonal based. And so the combination of these different kinds of signaling is what is thought to be underlying the capacity of development to create the diversity of the structures. Okay, enough with the introduction. Let's see at some published work that I'm going to go pretty quickly over as it is published. But just to give you a little bit of a sense of how we went about building what I consider sort of one of the first synthetic developmental programs. So in the lab of Wendelin and with Satoshi, we constructed these kinds of circuits where we had cell-cell signaling communication encoded with SynNotch pathways, and they were coupled with changes in cell-cell adhesion mainly, and or changes in color, or changes in having new signals expressed in this type of networks, and so then we developed a few of these signaling that had as output these morphological outputs and introduced them in fibroblasts, and then we were with the goal of introducing some sort of developmental trajectory in these fibroblasts. And we started with these fibroblasts that have this behavior. So these are fibroblast cells from a mouse. They grow in suspension. if you provide them with a non-adhesive surface. So this is probably the least exciting of the slides, but it's purposefully so because we wanted to start from a system that doesn't display any type of morphogenesis on itself. And then, so this is our blank slate, into which we start to introduce the artificial circuits, and then start to see some form of morphogenesis. So the first circuit that we introduced was having engineered two of these fibroblasts in two different ways. Sender cell that express a synthetic notch ligand, they're going to be blue, and a red cell that is a receiver that expresses an anti-CD19 SYN notch that has as a target gene icadirin. Icadirin is a adhesion molecule and used in again evolved during evolution that is expressed on the membrane of cells, and then when two cells express icadirin, they like each other and they stick together like in a Velcro-like fashion. And so with this circuit, we created two cells separately. We engineered them, sender and receiver, and we cultured them separately. And then at a certain point, we mix a little bit of the two in the same context that I showed you just a slide before of the normal cells. And then we do a time-lapse movie over 24, 30 hours. And this is what happens. The cells communicate that the icadirin is tagged with the GFP. So the red cells, show you again, the red cells turn green because they start to express the target gene of SYN notch when they are in contact with the blue cells. And not only they turn green, they also change their relationship with each other. The green cells start to clump and form the core of the structure, and the blue cell stays on the outside. So at the beginning, you had a random mixture. At the end, you have a two-layer structure, more organized. And so that was just the beginning, the simplest case that we thought of. And as you can imagine, as you can see also in the paper, by changing the receptors and the networks and the cadirins, you can achieve different structures. I show you here, I have a slightly more complex network in which you have sender cells activating icadirin on the receiver cells, and then the receiver cells activate back the sender cells through a GFP type of communication, and you are activating also cadirins on both sides. So the first step is the formation of this green core, and then the second step is the green core is signaling back to the blue cells to turn red and express the low level of icadirin. So this is what it looks like when you engineer the cells and then you mix them together. They form the green core, and then the green core communicates back to the blue cells to turn blue and to turn red, and it does so only to the closest cells, because again this is a contact dependent signaling. And there are more in the paper. One interesting feature of this building modality, which is this genetically encoded self-organization, is that it is dynamically encoded. The structure is dynamically encoded. It's not statically encoded. If you have an example of a statically encoded structure is a table. You build a table and it's rigid, it's there. And then you do some perturbation of the structure and the perturbation stays. So if you cut a table in half, the table stays. And you separate the two halves stay halves. Nothing really exciting happens. We did some experiment where we showed that our structures, if you cut them in half, they do something more interesting. They actually regenerate to the initial structure. And so this three-layer, we call this structure 3 layers, this green, red, and blue. We cut it in half, and then we let it we separate the two halves, and so we checked what happened in the two halves. And what happens is that slowly, over time, you go back to the 3 layers. And so this is more exciting than a table at this point, because it's like if you have a table that you cut in half and you take an half and half, the half of a table reconstruct the whole table. And so in this sense, it's superior building technology. And as I said, if you check out this paper, you see a number of other, structures generated with variations of these kinds of networks, variations of different adhesion molecules, and so on and so forth. Yeah, and so just to repeat, these fibroblasts are not adhering to the surface because the surface is treated in a way that makes it known that these fibroblasts don't like it. And so this is to maximize cell-to-cell interactions as opposed to cell-to-substrate interactions. Otherwise, you'd have, if you have a treated surface, the fibroblasts stick to the surface very strongly, and then the cell-to-cell interactions are not that important. What dominates the dynamics and the morphology is the cell-to-substrate. At least that was one thinking that we had behind the choice, and we like to see things in 3D. So these are a gallery of different structures that you can see in the paper that I cited before. What we are doing after that, we've been sort of motivated by the fact that we realized that the design of these circuits was done mainly in our brain and really through whiteboard, thinking, oh, wouldn't it be cool if we did this circuit? Oh, that's what should happen. And so it's sort of a limited, and then you try it, sometimes it works, sometimes it doesn't. And then you go back to the whiteboard. We thought that it could be an easier way, a faster way to explore different designs. If we could have a computational system that you can design a network and see what happens, that could be predictive of the things that you see in vitro. And so we have a bioRxiv now paper in revision that is doing that. That is doing the computational. This has done successfully, I would say. As you can see here, is an example of a prediction and an example of an in vitro result. We have a matching of the in vitro, the in silico structures and the in vitro structures. And what we're trying to do now is to try to push it a step forward and see if you can get much more elongated structures. So far, what we got in vitro, we got pretty much spherical or maybe ellipsoid, if you want, but nothing that really makes a long tube or a long body plan, an axial elongation. And so we are wondering if you can. We have been trying to find networks in silico that can do that, and so we have been successful in designing in silico some networks that would sustain axial elongation. And we're trying now to see if we can implement those in cells. And we have sort of one of the first results that's not the level of the computational system yet, but I've been happy to see it going from a completely spherical structure like this to a slightly more elongated structure if you use different cadherins again. And we are trying to combine the different cadherins with the different kinds of network where we have propagation of signaling to encode this elongation. Let me show you just briefly how the signal propagation circuit works. The signal propagation is on the bottom here. And it is an anti-GFP SIM notch that drives production of a reporter and cherry, and also a GFP ligand itself. So it's a positive feedback. The receptor activates the ligand for itself. And so if you have a sender cell, or in this case, you have a spheroid of sender cells. These are spheroid of 100 sender cells. And then you have a spheroid of 100 receiver cells. What you see is that at the beginning, the activation is at the interface, and then it progresses throughout the receiving spheroid. I'm going to play the movie, and this is what it looks like, the activation over time. play it again. So you have the sender cells are green and the receiver cells are turning red because they're passing a GFP signal along. And so far they're passing nothing else, just this green signal. And so at the end, the movie, still we don't get an elongation. But we are hoping to use these, and that is what is predicted by the computational system, that this would happen. What the computational system suggests is that if instead of passing just the red signal, you pass also a capacity of the cells to stop proliferation, to become more rigid in order to form more like a stalk type of structure, then you should be able to get an elongated structure. So we are working to replace these end-cherry signals with effectors that would make the stalk more solid, and we are hoping to get elongation. And the signal propagation circuit was developed by Marco Santorelli in the lab, and nowadays Josh is working on the implementation of the elongation. Yeah, so in summary for this part was to say that this is as far as we got in terms of morphogenesis and how controlling cell-cell communication with the synapse type of pathways, controlling output of the adhesion kind, and maybe of the fluidity kind. We can get some type of different morphologies. We can get some novel types of arrangements, and we can really start to control patterning and morphogenesis. And so that's exciting for us, as you want to establish a body plan where you can say then, okay, this side is going to be the head, this side is going to be the tail, the left or right is it's going to be an outer shell of endothelial cells, the inner shell, it's going to die and it's going to form a tube. So you want to first establish a geometry of patterning of gene expression, and that's what you see during development. There's a first phase where you start from a uniform population of cells that are sort of pluripotent or equally potent, and then you have a patterning. Certain cells take on certain fates, and so that we are at that stage in terms of creating different patterns of gene expression. What we want to do then is couple these with differentiation, and with different color of the structures becoming different cell types. As so far, there's just the cell is red and cell is green, but nothing really functionally really happens. So what we want to do is turning these red cells into something and the green cells into something else to do to go towards the functional differentiation. So that we started playing with. Sorry, this is again another part that I wanted to go over in a different context. So in this context, I wanted to talk about the differentiation, how we do differentiation. So differentiation is this process by which you start from a pluripotent cell or a stem cell of some kind, and you go down a differentiation path to become either a differentiated cell. It can be a neuron, it can be a muscle cell, an endothelial cell, a blood cell, and the progress is this happens at the level, let's say, of a single cell. You can look at a single cell, and these cells start to express different genes, becomes a progenitor of a certain kind, and then start to express the special functional genes that are specifically expressed only in that cell type to make, for example, the long fibers of actomyosin, contraction for a muscle cell. That the neuron cell doesn't need to express. And so it's through this form of progressive restriction of potential and progressive sculpting of the gene expression is you achieve a differentiation. And there's been a lot of works recently to show that you can what this is called a Waddington landscape, from the person that has developed the idea of seeing cells as beads that are going downhill in a landscape that's sort of rugged, and finding sort of wells of potentials that are these final stable adult cell fates. It was, when it was developed, the idea was that you go from up here, from the top of the hill, you go downhill, and that was the only. And then you can have different paths downhill. You can get a neuron, you can get a fibroblast, and that's it. That was just trying to understand how the different ways downhill leads to different cell types. What has been shown recently in the last 10, 15, 20 years is that the landscape is much more, can be traversed in a much more free way. You can move from one adult cell of one kind to a different kind, or you can go from a differentiated cell back to a pluripotent cell. So you can go back uphill from the differentiated cell type. And so this, the switching of one cell type to another cell type is called transdifferentiation. You can go from a muscle cell to a neuron even with certain protocols. And instead the going back is called inducing in pluripotent cell phase or reprogramming, in which you go from an adult cell to a stem cell. And this is one of the examples that has been shown is that, and so the trick is how. It's okay, how are you going to do that? For doing that, there are different protocols with growth factors, or initially, especially, it was shown that you can force overexpress certain genes, certain master genes for a certain fate, and achieve these movements, these non-natural movements in the Waddington landscape. So one of the first examples with this MyoD that's shown here, that if you, MyoD is a transcription factor, so it's a gene for a transcription factor. If you insert that into a fibroblast, cell of for example, of your skin, you take the cell of your skin in the culture, you insert MyoD, and now it starts to become a skeletal muscle cell. So we were, of course, excited about seeing that you can do differentiation with just one gene, for example. And so for us was the first proof of principle for us to convince ourselves that we could be able to do synthetic differentiation, was to put a synthetic notch pathway as a target gene of a synthetic notch pathway having MyoD. And so we build this cell with an anti-CD19 notch that drives, instead of driving just GFP, instead of driving icadirin, in this case, it drives MyoD GFP. And so and then we did experiments where we cultured those cells alone, or cultured those receiver cells with the sender cells that express CD19. And what's shown here is what happens when you cultivate them together, and you get to see already the differentiated myotube that has all these little circles of the nuclei of the myotubes, whereas the single cell looks more like this. One on the top, on the thing. Moving. Do you see my cursor? You did. And so these are the cells that haven't fused yet. But these are how the cells would look like if they weren't in contact with this CD19 ligand. And so this was for us very exciting. What was showing us that we can induce functional differentiation with SynNotch, that the expression of the target gene of a SynNotch pathway was strong enough to drive these type of behaviors. And so then we started seeing if we can incorporate them in those sort of patterning circuits, and we're doing that. And we're also trying to incorporate it in another kind of pathways that we are developing, and that are activated not from a neighboring cell, but they can be activated from a material. In these kinds of experiments, so far I've shown you that the signal for SynNotch can come from a neighboring cell, but we've also discovered that you can stick the ligand for SynNotch, for example, GFP, on a plate, and then the cells would read that signal from the plate and would activate a C-notch. So these cells that have a C-notch MyoD pathway will be activated can be activated from the surface. And so the exciting thing about being able to print to activate C-notch from a surface is that then there are ways to microcontact print the ligand in geometrically controlled fashions. So you can think that you can control the geometry of differentiation in this way. And we're doing this. The Meher is the leader in this project. There's a PhD student co-supervised by me and by a colleague, Megan McCain, in the biomedical engineering department that specialized in microcontact printing of proteins. And so what we do, Megan and Meher do their magic. They build stamps that they can use to stamp GFP on a plate, and then you get a surface where you can cultivate your cells that have GFP on stripes, for example, or you can have a GFP on a ring pattern or on a wavy pattern, or you can spell your name with GFP. And then what we do is those cultured surface, we put on our cells, our synthetic notch cells, that then read the ligand only when they see the ligand, and in between the lines they don't see anything, so they stay normal. And so these are an example of the results that we see when we see this is the first row is the GFP, so the ligand printed on the surface in a circle, in a ring pattern, or in rows. And bottom here is the activation of the target gene of the C-notch pathway. Again, in this experiment, the receiver cells are plated in the whole surface. The cells are everywhere. And they get activated in the target gene only in the exact place where the ligand is. So in a circle expression, on a ring expression, or in a row expression pattern, and these are the quantifications. And so that was shown as a proof of principle with sort of with only the target gene, only a reporter gene. But then we moved on to do experiments with the differentiation gene of MyoD, and so the hope was to not only see M-cherry, but to see really also differentiation in the place directed by the ligands, the synthetic ligands on the plate. And so these are the first sort of results that you can see it in the center. These big cords of cells is a myotube, whereas on the side you can almost make out, but they're just small, very small cells that are genetically identical to the ones in the center, but they don't see the synthetic ligand, so they don't do anything. And you see this with a circle, then the same cells with a different substrate, they do this, so they are activated on the rows. And where we are going with this is we are hoping to make it sort of multiple with multiple fates to control the geometry. With this, we are trying to see if the geometry of differentiation affects the differentiation itself. And sort of the next step is going towards a multi cell type differentiation, where we could have a synthetic cell, engineered cells with two C-notch pathways, one for skeletal muscle, one for endothelial, for example, and printing two ligands on the surface. And now you can decide the geometry not only on one cell type, but of a multicellular tissue. You can have muscles on certain regions, endothelial cells with specific geometry interspersed to achieve something that starts to look more like a native tissue. Okay, I think I'm doing my last recap before we open for question. I've showed you, I've skipped on the mechanochemical coupling, which I think might be more interesting as it becomes more developed. But I've showed you the contact dependent signaling, how controlling the adhesion can form some types of patterns in in a non-organizing system, and how we can control differentiation with this new way of doing ECM, or sort of scaffold to cell signaling. And I'm excited about sort of future directions of incorporating other receptors, as I mentioned, program growth of functional assemblies, and also collaborating for the organoids. And I just want to mention that I'm trying to develop some of these toy models as sort of teaching props. In a way, some of these models, like the 3-layer structure, for example, is a very simplified developmental system that one can use in teaching developmental biology. You're prototyping it with curriculum for high school, and see if, without any need to know anything about sea urchin, about the model system, which is super complicated, if just introducing a synthetic, simplified, can get the students to understand about signaling, about morphogenesis and patterning. Pretty much like when we start studying physics in high school, we don't study how a rocket ship is built, but we study a pendulum, or we study a very simple system. Just wanted to highlight this effort that I think is particularly exciting. As a close, I think this field is really new, really super exciting, and it's going to be we are doing sort of whatever we can in terms of the foundational ideas and putting out our first round of cool applications. But I think it's going to be the next generations. It's going to be you guys that are going to take on these and are going to make so much amazing stuff that I cannot even imagine. And so I'm very excited about hearing from you what you think is going to be the next cool things that can be done with this type of technology. With this, I stop it. I'm going to be available for any questions if you have.
Thank you so much, Professor Leonardo. Yeah, I really like how you're so excited for this field. So am I. So now we're going to start the Q&A session. So how it's going to work is basically you can either raise your hand and unmute yourself to ask a question, or you can type your question in the chat box. So I think I'll start with some questions from Olivia. I'll read out her question. So her question is, how does your work relate to Turing patterns?
Cool question. How much time do we have? Turing patterns. I can talk for hours about Turing patterns. It was just a joke. I can try to be brief. It's just want to say this. It's a great question. It's a big question. See, can I answer? So, Turing patterns, for the ones that don't know, are sort of Turing is sort of one of the inventors of the computer, if you want. So most like a mathematician, they put out a paper in the 50s where he was studying morphogenesis and saying, oh, if cells communicate in such and such way, you can get patterning out of noise. And so this one across the years, then it's been interpreted as being a specific class of patterns are called the Turing patterns these days, which are based on shorter range activation and long range inhibition. And so in this specific sense, we haven't implemented a Turing pattern with SynNotch. It could be doable, and we are thinking about how to do that. In a larger sense, a Turing pattern is anything that takes a uniform population of cells into differentiated into a pattern that's stable over time. In that sense, everything we do can be seen as a Turing pattern, especially as we start from uniform cells and how they can pattern themselves. So we are very much inspired by the notion of computational computing circuits that then endow cells or communities of cells with capacity of pattern themselves. That was sort of the shorter version of the answers. I hope I gave you something, Olivia.
Thank you. Olivia also asked a follow-up question. What do you think are the biggest modeling challenges for building multicellular systems?
There may name a couple. One is that we are trying to address is this design phase, which we are doing sort of whiteboard-like, and then it's slow, then you go back and forth. So we're trying to address it with this computational system. So that's one set of challenges. Another set of challenges is that sometimes cells don't behave like we expect them to behave, and so that to me shows that we don't understand the biology enough, which is both slowing down progress, but it's also exciting because it gives an opportunity to learn more about the systems. And one example is epigenetic silencing. There's just very briefly, epigenetic silencing is a process by which certain genes in the genome are sort of shut down permanently, even though they're there, they're sort of silent. And this is used all over the places during development to do what I said earlier, that select certain genes to be expressed in certain cells and certain others to not be expressed in certain cells. A challenge with when you introduce synthetic circuits in the cells is that sometimes they get silenced themselves. And there's this epigenetic mechanism. The cells doesn't know that these circuits are not supposed to be silenced because you don't want them, the cells, to silence them. But sometimes that happens, and there's very little way to control that so far. So that's definitely one big area of focus for the field. What you see is that your cells are doing what they need to do, but then after a while, the synthetic circuit that you've put in there's no GFP anymore. There's no synapse receptor expressed anymore, and it's really hard to revert. So far, there aren't that many tools. So that's definitely a challenge, especially for the more complicated kind of circuits that we want to put in. We certainly want to, if anything, we want to exploit this biological phenomenon of keeping certain genes on and certain genes off. But so far, we understand a lot of the epigenetic mechanisms. We haven't developed very precise tools, synthetic tools, to have a full control. So that's definitely something that will need to happen to enable the more complex circuits that can be deployed during a differentiation, for example.
Thank you. And I think building on that, I was like wondering, like how has that all affected the way that you're modeling these in like computer modeling?
Great question. So you're thinking about the epigenetic silencing specifically?
Yeah, just that, and also just in general, like I found it really interesting that you were combining both computational approaches and like in vivo stuff. So I was wondering, yeah, what challenges have you faced?
Something more there. So the one set of challenges with the computational system is that there's a lot of computational systems out there to model developmental systems. This is something that has been done for years to model search in development or mechano transductions. And the problem with those systems, these are very complicated systems, and it's not clear how you go in and just change a small parameter, and that can be changed in a way that you can then implement it in your system. They are done for analysis of a system. What we want is a computational system for doing design, which is more like what the engineers do. Engineers, they want to build a plane, they build it in a design software, in an AutoCAD, in a CAD type of design software, and then they go and then they spit out a recommendation for how to build it, and then they go and build it. So that's what we wanted to do, and one challenge was to simplify it enough and to make it so that whatever circuit is spit out as a recommendation is not a super complicated circuit with 100 nodes, but just with the nodes that you can actually then implement. So that was one set of things that we did. The other challenge that we are just starting to do is really combining having a sort of a closed loop with the implementation. When you start to have a prediction from the computational part, and you do a prediction, and then you implement in the cells. My overall prediction is that this is never gonna work 100% of the time exactly how you predicted, because we don't understand biology the way we understand mechanical engineering, and so it's and so it's going to be a challenge how you incorporate the feedback from the biological system when it doesn't work as you predicted, how you bring it back and change the computational system so that it's like, oh, okay, I thought it worked like X and it works like Y, and then I need to build a better system that predicts Y, because that's what, and so then refining the computational system with the biological feedback is going to be important, and it hasn't been done yet in this field. It's not exactly clear how to do it, and it's going to be exciting to go. I'm partnering with computational biologists, and it's definitely a frontier for the field. In certain for example, for genetic networks in bacteria, there are softwares that can do that. You can tell them what kind of genetic circuit, genetic output you want to have. The silo type of platform can predict the genetic network, exactly the genetic network that will give you that specific pattern of gene expression. But that really works for bacteria and for transcriptional networks. And so we want to sort of learn from that experience to do it in the mammalian cells version and the multicellular version, and there's morphogenesis. There's several layers of complications over that. See, there is another question.
Yeah, there is. I think to summarize, Francesco's question is basically, do you see a better way to a way to use these toy model systems aside from like teaching as like for teaching development or regenerative medicine? Like,
right.
Can you like add more layers of complexity and more features and to discover more things from it? Like, do you see that something that we can use the toy systems?
Yes, yeah. So I guess one application that I have hoping to get at is that we can get to build machines that they can interface with human biology in a way that we can help its regenerative capacities. Let's say we want to one sort of moonshot type of idea is the limb regeneration. If you have a limb amputation in certain animals, it forms a blastema of stem cells that direct the growth of a full limb. In humans, that doesn't happen. You can imagine that if you had a synthetic blastema, synthetic spheroid of cells that can start to orchestrate the development of a full organ, that's something that in principle is possible. And so having this synthetic system, multicellular system, interface with degenerative system in the human biology, I think it's an exciting interface. And even then, we were talking with surgeons, they say, oh, if you could build a certain structure, would be super exciting for us. We want a tube that long, that thick, with cardiomyocyte on the outside and endothelial cells on the inside. We want to, because we need we use a plastic tube right now. We want to use something that's more biological. If we can use our system to build something like that from patient cells, that would be exciting. It's an exciting in theory for us to think about how to do that, and then it could have applications in sort of regenerative medicine in that sense.
Yeah, I am so excited for that too. And I think just to wrap up with one last question, I was wondering how close are we, do you think, to like having regenerative medicine like that being a real thing?
Right, close. Yeah, that's an interesting question. They say you sort of under predict, you sort of overestimate what you can do in the short term, but you tend to underestimate what you can do in the long term. So, and I don't know what's short term and long term here. How far we are, I don't know. I'm pretty excited. I think we can do a lot of progress. You know, limb regeneration can take some 50 years, easy. Some more close applications can happen within, I think, within my lifetime. And this is also interfacing with a lot of progress that's been done in neighboring fields, like the organoids field or the tissue engineering field. They are doing they are already in the clinic. Some of those approaches are already used in the clinics. We just think that we can maybe help in some of those, and so that's also something that I wouldn't be surprised if in the next generation of organoids there's going to be some synthetic biology in there too.
Yeah, thank
But
you.
the
Yeah.
prediction is always hard to do predictions, you know, in technology.
Yeah, I think definitely it's a very exciting time, and there's like tons of development in the intersection between synthetic biology and regenerative medicine. So yeah, so I'll now have to wrap up the Q&A session. Thank you to Francesco and Olivia for giving us your questions. And once again, thank you to Professor Leonardo for coming today to deliver your amazing talk. Like, I'm so excited by all your research, and I'm looking forward to more and more developments from you in the field. So yeah, I think I can see a couple of like clapping hands from the audience. So yeah, so thank you everyone also for joining us today. We hope that you found today's webinar insightful and inspired by his Professor Leonardo's research. We're gonna end this meeting now, so please do sign up for our newsletter and follow our social media on Facebook or Twitter, and to stay posted for future events. So yeah, that's all from me now. Enjoy the rest of your day, everyone. Bye-bye.